Alrighty, so we are going to talk about cellular respiration. Now, cellular respiration is indeed one of the most fascinating metabolic pathways in many organisms. But at the same time, it's also one of the most complicated. A lot of students have trouble with cellular respiration and photosynthesis because they are uh, very not energy, material rich subjects. There's a lot of concepts to cover, and then a lot of stages or processes that occur in each one that need to be talked about. So there's a lot of material that's covered, and it can be very overwhelming for students. It's also easy to get them to confused with one another because they are similar in certain ways. I remember when I was going through high school in AP Bio, I found both to be very overwhelming. Again, because there was so much information and material to learn all at once. And we talked about cellular respiration and then moved immediately into photosynthesis, which for me, again, they are so similar in certain ways that it was difficult at times to keep them separate from one another. I was sometimes getting them confused. So taking that into account and kind of using the method that I ended up using to better learn or better understand the material that was presented to me, I'm going to break it down into kind of individual components, right? Going along the same concept of how do you eat an elephant one bite at a time. So you're going to see this slide pop up from time to time throughout this lecture, but it serves as a roadmap for how we are going to move and progress through the material for cellular respiration. And there's a ton of material to cover, even in this lecture, which is a condensed version of much of the information that exists, as well as many of the other lectures that you see. They're all condensed versions, but even so, they are still very rich in the material that's presented. So use this slide to help guide your understanding as far as where we're moving, how everything ties into one another, and also use it in case you end up or you feel like you're starting to get lost in the material. And this is kind of how I broke my notes down way back when I was a wee little thing in high school, and I felt that it worked well for me. So hopefully you can find benefit from it as well. So to start, let's actually look at what cellular respiration is, where does it occur, and what does it involve. With energy, right? Energy itself cannot be created or destroyed based on the second law of thermodynamics. And because of that, organisms must obtain their energy from the environment. Now, this energy that they gain is usually comes in the form of either food or solar radiation, depending on whether you're a plant or an animal. Now, the process of converting energy into a usable form is called metabolism. We had talked about that back in the energy and metabolism lecture. With regards to cellular metabolism, there are two methods that are most frequently used by different organisms. One is cellular respiration, the other is photosynthesis. Right, so we're going to look just at cellular respiration in this lecture. Photosynthesis is going to come later in a different lecture. So what is cellular respiration? Well, or what happens in cellular respiration? Cellular respiration converts the energy that's found in food molecules into a usable form of energy. And that energy being ATP, or adenosine triphosphate. So in other words, cellular respiration breaks down stored energy that's found in organic fuel sources or food into a usable form of energy, that usable form being ATP. And then that can be utilized for cellular work by the organism. Now, what can be used as fuel in respiration? Well, carbohydrates, fats, proteins, all of them can be used as fuel. When all three are available in the cell, carbohydrates will be used first, followed by fats, and then proteins. The reason why carbohydrates are used first is because they provide a much higher yield of ATP per molecule of glucose or any monosaccharide that is broken down. You'll learn about lipid and protein respiration or oxidation more in any biochemistry classes you may take later in life. For the purpose of this little lecture talk, I'm just going to focus on how glucose is used. Right? So we're only looking at one of your monomers for the carbohydrates, but it's the most important one. It's the one that's kind of the best energy source for cells. So we're just going to focus on the oxidation of glucose. In other words, how glucose is broken down to provide energy or fuel the synthesis of ATP. Now, you can think of the whole process of cellular respiration almost as like drawing money out of your bank account. You can take storage carbohydrates like starch or glycogen and think of them as acting as a savings account. And they're more specifically a savings account for chemical energy. And in this case, your ATP, on the other hand, is going to be the cash. Now, in order to withdraw chemical energy from these savings accounts to get your cash, which is ATP, something has to first be done to those storage carbohydrates, right? More specifically, in this case, the storage carbohydrates are going to be broken down into their individual glucose monomers. And then those glucose monomers are going to be used to produce ATP, either by cellular respiration or fermentation. Cellular respiration deals with respirative properties or processes that are occurring in aerobic conditions. Fermentation are processes that occur in anaerobic conditions. So the two differ based on the degree to which glucose is oxidized, as well as that environmental condition. You can learn about fermentation in the other lecture that is combined in this series. Again, we're just going to look at aerobic cellular respiration. Now, a mistake that or trap that some students fall into is making the mistake of thinking that cellular respiration is the same thing as systemic respiration or our pulmonary function. And they are not. They are not the same thing, but they are interrelated with one another, right? So when we breathe, we take in oxygen. When we eat something like pizza, we get carbohydrates. Now, the carbohydrates that we ingest from the pizza are broken down into glucose monomers through the processes of digestion. Now, both these glucose monomers that we have gained from digestion, as well as the oxygen molecules that we have breathed into our body, they are both going to enter our bloodstream and be carried to the body cells where cellular respiration occurs. And then carbon dioxide and water will be released as glucose is broken down in mitochondria, and that provides the energy for ATP production. So again, don't make the mistake of thinking that they are one and the same thing. They are not, but they are interrelated. Same holds true for any living organism, like this little ocelot friend here. Now, the major site, or where the majority of cellular respiration is going to occur, is in an organelle called the mitochondria. The mitochondria is known as the powerhouse of the cell. This organelle has membranes that are specialized for aerobic respiration. The matrix of the mitochondria will serve as the site for the Krebs cycle. And then the compartments that are created by the foldings of the inner membrane of the mitochondria will serve as the site for the electron transport chain and the majority of ATP synthesis. And again, we'll talk about each one of those processes more in depth a little bit later. All right, so that kind of is the intro to cellular respiration. Right? Now you know what it is, why it's occurring, and primarily where it actually is occurring in the cell. Now let's talk about what the products are of cellular respiration. Obviously, the major product is ATP, but there are actually three important products, right? So ATP, yes, is one. It's one of the major products. But there are also two high energy molecules produced that serve as electron carriers. And those are NADH and FADH2. So let's talk first about ATP, right? ATP you can think of as being the cell's energy shuttle, or the related to being kind of the cellular energy currency. ATP stands for adenosine triphosphate. Its internal structure is, if you look over here, is actually an RNA nucleotide, right? Because it has the ribose sugar that's connected to three phosphate groups, or has three phosphate groups attached to it. Hence the reason why it's a triphosphate molecule. It's called adenosine triphosphate because it has the adenine nitrogenous base attached to the ribose sugar. Now the bonds that are formed between these phosphate groups are high energy bonds, right? That means there's a tremendous amount of energy that is stored, or potential energy, within each one of these bonds. You have an alpha bond, beta bond, and a gamma bond. So when those bonds are broken, that potential energy that is stored is released. So you can release a tremendous amount of energy from the breakage of these bonds in ATP, which is why ATP becomes your energy currency. So as far as its purpose in storing energy, right? Again, the energy will be stored in those bonds, but only for a brief amount of time. ATP itself is not a long-term source for storing energy, again, long-term. It'll allow the cells to store energy briefly and then transport it within itself to support other chemical reactions or cellular work. So as ATP is used for energy, when it's accessed for that, a phosphate group from the three amigos here will be detached, right? And in doing so, that releases energy. That energy is used to fuel or support different endergonic reactions. When you also have the break off of that phosphate group, ATP is converted into a form called ADP, meaning it only has two phosphate groups. So it's adenosine diphosphate. So the breakdown of ATP creates ADP and an inorganic phosphate molecule. The energy that's derived from glucose catabolism, or the oxidation of glucose, will be used to recharge ADP. Right? So this will be used to regenerate ATP by linking ADP with a phosphate group, or recharging ADP. Now, as I said before, there are two energy-carrying molecules that are also products of cellular respiration. The first one that we'll talk about here is nicotinamide adenine dinucleotide, or NAD+. And there are two forms that we're going to talk about, NAD+ and NADH. Right, so what's the difference? Well, one is what we call oxidized, the other is reduced. We'll talk about what's actually meant, or how these processes work as far as oxidation and reduction in a little bit. But essentially what's happening is one form is losing electrons or a proton, while the other form is gaining those. So they are essentially the same compound per se, just different forms. Again, one is oxidized, the other is reduced. So the reduced form of NAD+ is NADH, right? If NAD+ is reduced, that means it gains a proton or gains electrons, and in doing so, it's converted or reduced into NADH. Now, NADH, likewise, can be converted into NAD+. NAD+ is the oxidized form. So it is the form of NADH that has lost a proton or has lost electrons. So when NADH loses electrons, it's converted into NAD+. When NAD+ gains protons or electrons, it's converted into NADH. Now, this whole kind of back and forth process between an oxidized form and reduced form occurs via enzyme, right? And this enzyme is called dehydrogenase, and you can pretty much tell its function based on the name. So whenever you have some compound that ends in ASE, you know that it's an enzyme. So obviously dehydrogenases, okay, so it's an enzyme. What is this enzyme doing? Well, dehydrogenase, that dehydrogen part, means to lose hydrogens, right? So it is removing hydrogens, or it's transferring hydrogens. And that's exactly what this enzyme does. The dehydrogenase will remove a pair of hydrogen atoms. So it'll remove 2 electrons and 2 protons from some substrate, right? So here's our substrate here. It's going to remove those 2 electrons and 2 hydrogens from the substrate. That enzyme, right, your dehydrogenase, will then add or deliver 2 electrons and 1 proton to the NAD+, right? So both of these electrons, 1 of these protons, are going to be added to the NAD+ by dehydrogenase to convert it into the reduced form, which is NADH. Hold on one moment. I think Figaro is eating something he should not be eating. What are you doing? What are you doing? No, that's a leaf. No, you're not eating the leaf. Can you not act like a 5-year-old for once? Just for like 2 seconds. I'm going to take that as being a no. Okay. So, so in that, by adding this proton and these 2 electrons, the dehydrogenase will convert NAD plus into the reduced form, which is NADH, or nicotinamide, in its reduced form. Now, this reduced form likewise can be converted into the oxidized form by the same enzyme. This enzyme will remove, or by removing 2 electrons and a hydrogen ion or a proton from your NADH, it can oxidize NADH into your NAD plus, again, which is the oxidized form of NADH. Same concept holds for the other electron carrier or high energy molecule that is created by cellular respiration. This one being flavin adenine dinucleotide, or FADH or FADH2, depending on the form that we're looking at. So there are 2 forms. You have FADH and FADH2. Some people will just refer to this as FAD. When I talk about it, I like to include this hydrogen so students aren't getting confused as to why you're suddenly going from FAD to FADH2. So we'll keep it this way. It also helps to separate this for cellular respiration when we start talking about photosynthesis. So this will act in the same way. FADH is the oxidized form. FADH2 is the reduced form. So FADH is reduced or gains electrons or a proton to form FADH2. FADH2 is oxidized or will lose the hydrogen, the proton or the electrons to form FADH. So those are the products. That way you know what these products are. You're also familiar with the oxidized and reduced forms for each of them, because again, that's going to come into play. It's going to come into play with what we talk about next, which are the reactions of cellular respiration. So again, I want to talk about these nitty-gritty reactions before going into the stages of respiration, because you will see these reactions occurring in each of these stages, and it's nice to know what each reaction entails and what's happening in each reaction prior to going into the stages. It allows you to better understand what's actually occurring in the stages and why by doing such. There are 2 major reactions that are occurring in cell respiration. One are coupled reactions, and the other are redox reactions. Now, coupled reactions, these are or refer to a system of reactions that are linked by some sort of energy shuttle. And in this case, ATP serves as an energy shuttle. ATP powers a lot of cellular work by coupling exergonic and endergonic reactions. And it's actually creates a process that is extremely efficient for the cell, especially in regards to energy consumption. So when we talk about forming ATP, ATP itself is regenerated or formed via the addition of a phosphate group to adenosine diphosphate, right? So ADP plus a phosphate group gives you ATP. Now, this is an endergonic reaction, right? So that means it requires energy. So energy has to enter the system, right? That's how I say to remember that it's endergonic. And because it requires energy, it's not spontaneous. So energy must be spent in order to create ATP. But unfortunately, a high amount of energy is needed to actually synthesize ATP. So where does this energy come from, first of all? And how can this process be made so that it is energetically efficient for the cell? And that's where the beauty of this comes in. The answer to that is through coupled reactions. Free energy that is used to phosphorylate ADP, meaning the energy that's used to add this phosphate group to ADP, or your adenosine diphosphate, actually comes from catabolic reactions that are occurring in the cell. Those catabolic reactions, ignore this here, this is incorrect, they are largely exergonic, right? So energy is being released by these reactions. They are energy yielding processes. The energy that comes from these catabolic reactions will be coupled with ADP and your phosphate in order to link them together to synthesize ATP. So by using energy that comes from these catabolic reactions, it prevents the cell from having an energy deficit, right? It prevents the cell's own energy from having to be used. Again, it's more energetically efficient. So you kind of have this shuffling, per se, of inorganic phosphate, ADP, and energy from catabolic reactions. And together they form what's called the ATP cycle, right? So this beautiful cyclic process that occurs that deals with the linkage of your catabolic and anabolic, or your exergonic and endergonic reactions together via ATP. So again, your energy is released from catabolic reactions. That energy is used to synthesize ATP. ATP can then be broken down to release the potential energy that's stored in it to drive cellular work, right? So the breakage of, or breakdown of ATP can then be used to drive or fuel endergonic processes, right? So again, it's very much like the coupled transport that we talked about with secondary active transport, where the passive diffusion of one molecule generates the energy that's used to transport another molecule up its concentration gradient. It's, again, the same concept here. The energy that's released from an energy yielding process is used to generate ATP. So that's the cell is not losing or coming into debt as far as energy expenditure. And then that product of ATP can be broken down to release energy, and that energy can be used to drive cellular work or drive endergonic or energy consuming processes. So again, a very beautiful system or a cyclic process that allows for the production or synthesis of ATP in a very energetically efficient way. Furthermore, when we talk about kind of this cyclic process where you see the connection or linkage of exergonic and endergonic reactions, or catabolic and anabolic reactions, you also see the catabolic and anabolic pathways being linked due to the intermediates that are produced by cellular respiration. So not only are they linked via the regeneration of ATP, they're also linked based on some of the intermediate products that are produced during different stages of cellular respiration. So for example, acetyl coenzyme A, right? So this serves as the starting point for anabolic pathways that will result in the synthesis of your fatty acids. And then these fatty acids will go on to build phospholipids, or your fats. So you have this intermediate in cell respiration serving as a precursor molecule for anabolic reactions, right? And again, you see this for different reactions that are leading to the synthesis of carbohydrates, nucleotides, amino acids, lipids, right? So this was just one example with some of your lipids. Another is looking at the intermediates from glycolysis, right? So the intermediates from glycolysis can actually be used to synthesize the substrates for nucleotide synthesis, right? These intermediates will synthesize or can be used in the synthesis of ribonucleotides and deoxyribonucleotides. And then those nucleotides in turn will be used as the building blocks for RNA and DNA synthesis. So again, you see this cyclic process, how everything is interconnected in a way that one molecule can serve different functions in the cell. Again, this is just another element of efficiency and energy saving. Now going back to ATP as far as its production, or how we generate ATP, there are three ways that ATP is generated by the cell. We will be seeing these two methods in action when we go through cellular respiration. Photophosphorylation is specific to photosynthesis. So this is the generation of ATP via the energy that's released from the transfer of electrons between chlorophyll and channel proteins. So we won't see this in action with cellular respiration because cellular respiration does not deal with the light reactions and impacts on chlorophyll. So you'll only see this with photosynthesis. But these two we will see in action when we talk about cellular respiration. So this is one way to generate ATP. The other two, again, the ones that we're going to focus on, are substrate level phosphorylation and oxidative phosphorylation. So substrate level phosphorylation, this just deals with the transfer of a high energy phosphate group to adenosine diphosphate. And that transfer of that phosphate group to ADP creates or synthesizes ATP. Oxidative phosphorylation, this deals with the transfer of electrons between two compounds. So it deals with redox reactions. One compound is gaining electrons or losing electrons, right? So one is losing, the other is gaining, or one is gaining, the other is losing. In other words, one is being oxidized, the other is being reduced. And that transfer of electrons between those two compounds releases energy. And that energy is then harnessed to generate ATP, specifically through a process called chemiosmosis, which we'll talk about at the end of electron transport chain. So that's looking at your coupling reactions. And those deal primarily with cellular respiration as far as generating ATP. The other major type of reactions in cellular respiration are called redox reactions, which I've been hinting at a little bit throughout the lecture up until this point. So in many chemical reactions, there is a transfer of electrons from one reactant to another. And these transfers are called oxidation reduction reactions, or redox for short. So the loss of electrons from one substance is called oxidation. The gain or addition of electrons to another substance is known as reduction. You can remember it by the acronym OIL-RIG. So oxidation is loss, reduction is gain. Right? So again, if a substance is losing electrons, it's being oxidized. If it is gaining or adding electrons, it is being reduced. Now, in these reactions, you always have one substance that is serving as the electron donor, the other that is serving as kind of the electron acceptor or recipient. So one is always being oxidized, the other is always being reduced. In this reaction up here, looking at your X and Y, in this reaction, substance X is the electron donor. It is becoming oxidized, so it loses its electrons, and therefore serves as the reducing agent. Right? We say that it serves as the reducing agent because it's going to end up reducing Y. Y is going to pick up those electrons that were lost by substance X. Right? So X is losing the electrons, Y is accepting or gaining those electrons. So X is becoming oxidized, Y is becoming reduced. Right? So because it's becoming reduced, or because Y is being reduced, it serves as the electron acceptor. We also say that Y is, because it's gaining electrons, because it's being reduced, it is the oxidizing agent, because it will oxidize X by removing the electrons from X. So the substance that is being oxidized is known as the reducing agent. The substance that is being reduced is referred to as the oxidizing agent. If that doesn't make sense, just throw some comments in the comment section. I know that, again, that can confuse a lot of people, but maybe by seeing it actually occurring in glucose, it'll make more sense. So let's look at the actual process of oxidation of glucose. So here, glucose is being oxidized, right? It's electrons are being lost from glucose in order to convert glucose into carbon dioxide. So because glucose is losing these hydrogen atoms, it is being oxidized. Now, glucose itself, even though it's being oxidized, we call it the reducing agent, right? Because it will give those hydrogen atoms that are lost to the oxygen molecule, right? So again, glucose is being oxidized. Because it's being oxidized, it's the reducing agent of oxygen, right? It's going to be the agent that causes the reduction of oxygen. So oxygen in the same process is being reduced. It's gaining those hydrogen atoms from glucose in order to create water. So because it's being reduced, again, it's gaining those hydrogen atoms, we call oxygen the oxidizing agent. Again, it's the oxidizing agent because it is receiving or gaining those hydrogen atoms from glucose. So it's causing or fueling the oxidation of glucose. Glucose is supporting or fueling the oxidation of oxygen in this case. So those are the two major reactions. Again, redox reactions we'll see continuously occurring throughout these different stages of cellular respiration. Now, a lot of people will refer to 3 stages of respiration. I break them into 4 because I include the intermediate stage, which deals with the oxidation of pyruvate as a separate named stage. Again, it just helps break it up for students a little better. So in all these processes, you have the atoms of glucose and oxygen taken and rearranged to create carbon dioxide and water and energy. And then that energy is used by the cell to produce ATP. Now the cells will produce ATP from glucose through these 4 interconnected processes or stages. Right? These stages being first glycolysis, followed by the intermediate or transition stage, which is the oxidation of pyruvate, the Krebs cycle or citric acid cycle, and oxidative phosphorylation, which is the electron transport chain and chemiosmosis. Now, the first 3 processes deal with the oxidation of glucose, and they will produce NADH and FADH2. And those carriers will then be fed to the electron transport chain. Now, again, we're going to delve into each one of these stages individually. And as we do that, I want you to keep asking yourself 3 questions and to organize your notes in such a way. 1, what goes in? 2, what comes out? And 3, where does each step occur? So looking at, again, what goes into the system. So what are the reactants of the process? What comes out or what is produced by each process? And where does each one occur in the cell? Again, the majority of them are going to occur in the mitochondria, but not all of them. And some of them may be different for eukaryotes versus prokaryotes. So we'll start with the first stage of cellular respiration, which is glycolysis. Glycolysis will occur in the cytoplasm or the cytosol for both eukaryotes and prokaryotes. Now, this process converts 1 6-carbon glucose into 2 molecules of pyruvate. And pyruvate is a 3-carbon compound. And this compound will then be used later to go through a conversion that's then used to create a product for the Krebs cycle. But during this process, ATP is produced via substrate level phosphorylation. So it's produced from ADP and an inorganic phosphate being linked to one another. And also during this process, NAD plus is reduced to form NADH. Now there are 2, well, before I get into that, important thing to remember with glycolysis is that it does not require oxygen. So glycolysis will occur whether or not oxygen is present. So it is seen in both aerobic and anaerobic cellular respiration, as well as fermentation, which is unlike the other processes. The other processes that we'll talk about for cell respiration require oxygen. But with whether it's aerobic or anaerobic, you can have glycolysis occur because it does not require oxygen. Now, as I was trying to jump into before, there are two major phases, per se, of glycolysis. We break it into two components or parts. The first is an energy investment phase, and the second is an energy payoff phase. So the first phase is this energy investment phase. And it's named aptly so because it requires the investment of energy initially. In this phase, it kind of prepares the 6-ring carbon of glucose for separation into those 2 3-carbon sugars, which are the pyruvates. Now, again, in order to do that, energy from ATP is invested into the molecule in order to energize that separation. We won't go through each nitty-gritty step. Again, the purpose of this is to give you an overview, somewhat complex overview of each process, but I won't ask you to memorize each step of each process. But essentially what's happening is you are breaking the glucose molecule down into smaller and smaller carbon compounds, right? So in the first stage, again, you have to invest 1 ATP molecule to allow for this glucose to be phosphorylated to form glucose 6-phosphate, which is this same 6-ringed glucose molecule, but it has an additional phosphate group attached to it. Following that, a second molecule of ATP has to be added to allow for a second phosphate molecule to be added. So now you have your essentially a glucose with 2 extra phosphates attached to it, or more specifically, a compound that's called fructose 1,6-biphosphate. 1,6 is just telling you where these 2 phosphate groups are attached, or what carbon. So it's attached at the first carbon and the 6th carbon. And then again, you have to have ATP invested in order to allow for the creation of this molecule. From here, we go into the energy conserving stage, right, which is the second phase. It's the energy payoff. So this half of glycolysis extracts ATP and high energy electrons from hydrogen atoms and attaches them to NAD+. Right, so after right at the end of the energy investment stage, you cleave this molecule in half so that you get 2 3-carbon compounds, each containing a phosphate group. Right, they're each called DHAP. And then those are rearranged to form 2 molecules of G3P. Now these molecules have the addition of 2 phosphates, and also kind of that addition is fueled by the reduction of 2-NAD+, to continually, it adds another phosphate group. The phosphate group that's added is very unstable because it's so high energetically. So these will almost immediately break off. And the break off of these phosphate groups allows for the regeneration of ATP through substrate level phosphorylation. So it forms 2 ATP. Now you have 2 separate compounds, each being, again, these 3 carbon compounds attached to a phosphate group. You extract or remove a water molecule, and that rearranges those compounds to a position that puts those phosphate groups in a less stable position. So they're higher, they're more high in energy, which allows them to be broken off or cleaved to release energy that again is used to fuel the substrate level phosphorylation of another 2 ATP molecules. And what you're left with is 2 molecules of pyruvic acid. Right. So in summary, with glycolysis, initially you have 2 ATP molecules that are invested in the first half. Now, overall, 4 ATP molecules are formed. Right? These 4 ATP molecules are formed during the second half or the energy conserving stage of glycolysis. But because we used 2 molecules of ATP in the first half, the net gain of glycolysis is only 2 ATP, right? So talking about net gain as far as the products of glycolysis, you have the formation of 2 ATP and 2 NADH. You also have the release of water in the reaction. And then the major product that you're left with that will move into the next stage of respiration are your 2 molecules of pyruvate. Right? Pyruvate is the 3 carbon compound that's created during glycolysis. So for every 1 glucose molecule, you get from glycolysis 2 ATP, 2 NADH, and a water molecule. This, again, is not looking at the net gain. So remember, because 2 ATP were used at the start of the process, you have a true net gain of 2 ATP, even though 4 ATP are used. All right, so where do we go from here? Well, we go into the second stage of respiration, which is the transition stage or the intermediate stage, also known as the oxidation of pyruvate, because that's exactly what's happening in this stage. In this stage or state, the pyruvate is being oxidized through a series of processes to create a compound called acetyl coenzyme A. Now, this whole step is carried out by a multi-enzyme complex, and it will catalyze 3 reactions to bring pyruvate into the mitochondria. Right? So remember, glycolysis is occurring in the cytosol. Right? So immediately following glycolysis, you have 2 pyruvate molecules that are just hanging out in the cytosol, and somehow they need to get from here, point A, to here, point B. So they need to move into the mitochondria. That's going to be done again by these 3 reactions that are carried out by the multi-enzyme complexes. So the pyruvate that's produced from glycolysis, again, is transported from the cytosol to the mitochondria. And it will move across the mitochondrial outer membrane through small pores and is then transported into the matrix by carrier proteins. So you can see your little pore or your outer membrane channel, and then the symport protein that will be used to transport or move the pyruvate from the outer membrane into the mitochondrial matrix. So first, you start off with 2 molecules of pyruvate. Those 2 molecules of pyruvate are oxidized, and they are transformed into molecules of acetyl coenzyme A. Now, the process or the reaction itself that is occurring, this first one is occurring across the mitochondrial membrane, will produce 1 NADH per pyruvate. Now remember, the conversion of glucose in glycolysis results in 2 pyruvate molecules. So because you have 1 NADH that's produced per pyruvate in this first step, that means there is a total of 2 NADH molecules that are produced during this transition stage, right? Because you have 2 pyruvate molecules that are made or produced from glycolysis. They are produced from 1 molecule of glucose. From 1 molecule of pyruvate, you get 1 NADH because you have 2 pyruvate molecules produced, and going through this reaction, you get 2 NADH. So here you have 2 NADH per glucose, again, because you have 2 pyruvate that are being converted. So the reaction converts 2 molecules of pyruvate into 2 molecules of acetyl coenzyme A. So that's acetyl coenzyme A is the major product. Again, you're going to have 2 of those. What's going into the system is your 2 molecules of pyruvate. So the products are going to be your 2 molecules of acetyl coenzyme A. You're also going to have the production of 2 molecules of NADH. Remember, there's 2 molecules that are NADH produced because there's 1 that's produced per pyruvate in this reaction. You're also going to have the release of carbon dioxide. Now, the acetyl coenzyme A that is produced here, or kind of arises from the transfer of an acetyl group to coenzyme A, that compound is going to move into the next stage of cellular respiration. So as I said, all of these stages are interconnected, right? So the transition state, glycolysis and the transition stage are connected via the production of pyruvate. The transition stage is connected now with the third stage via the production of acetyl coenzyme A. So acetyl coenzyme A is going to move into the third stage, which is known as the Krebs cycle, or the citric acid cycle. Now, the Krebs cycle occurs in the mitochondrial matrix. This was a process that was discovered, by a man named Hans Krebs, which was a German-British scientist back in the 1930s. And he discovered this process when he went on this dogged pursuit to solve this puzzling observation with carboxylic acids in cells. Now, at this time in history, there were 8 carboxylic acids identified by biologists that could be rapidly oxidized. And the redox reactions that involved these 8 carboxylic acids often produced carbon dioxide, which is the endpoint of glucose oxidation via cell respiration. Now, the thing that was causing an issue was the fact that when biologists added any one of these 8 carboxylic acids to cells, the rate of glucose oxidation increased, suggesting that these reactions were somehow involved or connected to the pathways involved in glucose catabolism. Okay, so that's great. So now we know that these carboxylic acids are tied or have a direct correlation or involvement in the breakdown or oxidation of glucose. But this is the weird thing, right? So whatever carboxylic acid was added next, right? It didn't matter which one. It didn't appear that it was used up or consumed in the reaction. Instead, all of these acids would later be recovered. So what on earth was happening that allowed these carboxylic acids that were supposedly consumed in these reactions to actually reappear? That's where Krebs came in. So Krebs proposed that these 8 carboxylic acids were connected to one another in a cyclic sequence rather than a linear pathway. Right? At this point, up until this time, everybody was trying to deal with this situation by looking at these reactions on a linear table, per se. Krebs said, let's take a step back. Maybe it's not a linear process. What if they are connected to one another in this cyclic feedback-like process to create a cyclic pathway? He also suggested that the reaction sequence itself was directly tied to the processing of pyruvate. Right? So at this point, we didn't know how they were connected or didn't even think of them as being connected. And his method of thinking turned out to be correct, and it created what we now have as the Krebs cycle or the citric acid cycle. And it's aptly named because he discovered it, but also because it starts with citrate, which is the salt of citric acid after you have the release of protons. So that's kind of the background story of the Krebs cycle. Again, I'm not going to expect you to memorize each stage of the citric acid cycle. It is something that you should know, but if you're in my intro bio course, I'm not going to make you memorize it. If you're in one of my more advanced biology classes, it's going to be a different story. So you'll be getting a nice acronym for that. But in this video, we'll just kind of give the nitty-gritty summary. So up until this point, right, you have glycolysis, which is the oxidation of one glucose molecule to produce 2 pyruvic acid molecules. And then those 2 pyruvic acid molecules go through the transition stage where they're oxidized to form 2 molecules of acetyl coenzyme A. Again, that acetyl coenzyme A is going to enter or be the molecule that's entering this Krebs cycle. Again, tying in the transition stage to this 3rd stage of cellular respiration. Now, the energy that's released by the oxidation of acetyl coenzyme A, which is going to be kind of the first step of the Krebs cycle, is used to ultimately produce 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of ATP, or an equivalent of ATP. In some cases, you may have, instead of 1 ATP being produced, you'll have 1 guanosine triphosphate, or GTP, produced. And this depends on the version of the enzyme that is used in the here in the fifth step of the Krebs cycle, as well as the type of cell that this is occurring in. Some multicellular eukaryotic cells express an ATP-specific succinyl coenzyme A synthases. So they operate on the succinyl coenzyme A, but there's others that express a GTP specific. So if it's an ATP specific synthase, then you will get the production of, or the substrate level conversion occurring here to form ATP. If it's GTP specific, instead of getting one ATP here, you'll get the synthesis of one GTP. In this case, we'll just look at ATP, but they are equivalent. Now, because the oxidation of pyruvate from the transition stage produces two molecules of acetyl coenzyme A, we're going to have two turns of the Krebs cycle. So the Krebs cycle will turn twice per one molecule of glucose that is processed in cellular respiration. And it turns twice again because from that one glucose, you have two pyruvic acids produced, which produce or are transformed into two molecules of acetyl coenzyme A. Because two molecules of acetyl coenzyme A are being added or moving into the Krebs cycle, you'll have two turns of the Krebs cycle. So for each turn of the Krebs cycle, again, you're going to have 3 NADH, 1 FADH2 molecule produced, 1 ATP, and also 2 molecules of CO2. Carbon dioxide is also going to be a byproduct of the Krebs cycle. Again, because you have two turns, the kind of net gain or the net total of the products that are produced are 6 NADH, 2 FADH2, 2 molecules of ATP, and 4 molecules of carbon dioxide. Now, up until this point, what's the total number of ATP we have? Well, we have 2 ATP total coming from here. We had a net total of 2 ATP from glycolysis. So at this time, at the end of the Krebs cycle, or by the end of the Krebs cycle, there's 4 ATP that have been produced. And we can look a little bit at kind of what's going on in some of these steps. If you look at them again, you see these very familiar redox reactions, right? So that's how we're getting the generation of the NADH and FADH2 molecules, which will serve as your electron carriers that will come into play during the electron transport chain. So kind of to start, during the cycle, each acetyl coenzyme A is kind of goes through a transition reaction that involves the conversion of NAD plus into NADH, as well as the release of carbon dioxide. So here you'll actually have 2 carbon dioxide released, and then your NADH molecule. During the other steps, kind of going from here, so now you have your acetyl coenzyme A, it goes into the first step. The acetyl group that's carried by coenzyme A, again to form acetyl coenzyme A, will combine with a 4-carbon compound that's kind of the starting point here, called oxaloacetic acid. And when the acetyl group combines with that compound, it forms citric acid. Again, that's going to be produced kind of from this citrate, which is the salt that forms citric acid once it releases protons. So you'll also have kind of the joining of those carbon molecules. So at this point, when you have the joining of 2 carbon molecules with the 4 carbon molecules, it'll help to recycle the acetyl coenzyme A. From here, you go again into other redox reactions. So you have basically the conversion of different carbon-based substrates into smaller carbon-based substrates to allow for the release of carbon dioxide, but also by breaking those bonds or breaking down into smaller carbon molecules, it releases energy and electrons, which are carried off by NADH. So it allows for the conversion, or is used in the conversion of NAD plus into NADH. You also see the same occurring with FAD plus and FADH. FADH and FADH2, where the specifically occurring here at stage 6 between the succinic acid and fumaric acid. So you have the conversion of this carbon compound, which allows for the release of high energy electrons that are added to or accepted by FADH. So these electrons are released from the succinic acid. They are gained by FADH, which allows for the conversion of FADH into FADH2. Again, these are just the different redox reactions that are occurring. Again, I'm not going to, if you're in my bio class, ask you to remember each one of these carbon-based compounds or the different substrates that are used at each of the 8 steps of the citric acid cycle. My other classes, I will provide you with a nice fun acronym for to help you remember them. So in summary, again, you're going to have 2 turns of the Krebs cycle per glucose molecule that is added to the system. And in general, the Krebs cycle is a cyclic process that is used to generate more higher energy molecules, as well as produce additional CO2 molecules, which are the byproducts of this reaction. So for each turn, of the Krebs cycle, you get 2 carbons, which exit completely as carbon dioxide. You have 3 NADH and 1 FADH2 molecules formed, and 1 molecule of ATP or an equivalent that is made by substrate level phosphorylation. Again, that's for each turn of the Krebs cycle. Because it will make 2 turns, because there are 2 molecules of acetyl coenzyme A entering the system. That's the kind of what goes into the system. That's the 2 molecules of acetyl coenzyme A. You will get a total of 6 NADH produced, a total of 2 FADH2, a total of 2 ATP, and a total of 4 carbon dioxide molecules. So up until this point, kind of looking at what the overall totals are, right? We have 10 NADH molecules, right? 2 came from glycolysis, 2 came from the oxidation of pyruvate, 6 came from Krebs cycle. We have 2 FADH2 molecules, right, both of which came from the Krebs cycle, and a total of 4 ATP molecules, 2 having come from glycolysis, right? Remember, 2 is the net gain for glycolysis, and 2 from the Krebs cycle. So in the transition stage, a lot of people will kind of skim over that because it does not produce ATP. It is important because it does produce those 2 molecules of NADH, and it also produces the starting material that's important for the start of, or used at the start of, Krebs cycle. So these are our totals up until this point. Now what? What happens from here? Well, what's going to happen is these, your NADH and FADH2 molecules are going to move into the fourth and final stage of cellular respiration, which is collectively referred to as oxidative phosphorylation, and it's more specifically is comprised of the electron transport chain and a process called chemiosmosis. Now, these molecules these are your electron carriers. They are going to carry electrons and move themselves into the electron transport chain, where they will go through a series of reactions that allow for the conversion of these carriers to be converted or allow for the synthesis of ATP, right? So we're going from these molecules, basically converting them into molecules of ATP, right? Our actual cellular cache value. So doing that, we have to talk about the specifics of your electron transport chain, right? So how does this happen? How do we go from NADH and these FADH2 molecules to ATP? I'm actually going to stop this lecture here, and you can kind of just stay right where you are, not move, keep watching, and we'll go right into the electron transport chain, just so it kind of breaks up this lecture a little bit for you.